So today we are going to cover let's call it a theory, but really a theory is backed by a lot of evidence, and this is something that we can never get hard empirical evidence for because it's a proposal that supposedly happened many, many millions of years ago. The transition from single-celled organisms like bacteria into what are called eukaryotes that have a nucleus and organelles. And there's a theory proposed called the endosymbiosis theory or hypothesis. So we're going to take a look at how feasible that is. It's still widely regarded as what must have happened, but we're going to see that it actually cannot happen. So a little background on the theory. In order for cells to get larger and more complex, that's going to put a high energy demand on the cells. So the question is, where's it going to get all this extra energy from to keep growing and growing and be able to divide and take on more advanced features that require more and more ATP energy? Well, this theory postulates that maybe in the distant past, maybe a couple of times, one cell had completely engulfed another cell, and so you have this host cell that got larger while the sort of, not really parasitic, it's a symbiotic relationship, got a nice cozy home to live in, got lots of nutrients, and in return provided some extra energy so that the two of them could grow into a more complicated eukaryotic precursor cell through symbiosis, this mutual benefit where the host cell provides protection and nutrients, and that captured cell, the endosymbiont, generates the extra energy needed for the two of them to grow ever larger and more complex. This is first proposed by Lynn Margulis in a paper on the origin of mitosing cells in 1966. She's obviously evolutionist, but she actually criticized a lot of what Darwin proposed as this whole idea of the strong versus the weak, survival of the fittest. She saw nature as much more symbiotic, much more of a partnership than a competition. So she was critical, but obviously still thought that there was this long process of millions of years of getting simpler to more complex cells. It just wasn't as sort of competitive and bloodthirsty and parasitic as Darwin proposed. A little interesting tidbit, she was actually married to Carl Sagan. Anyone remember the astronomer Carl Sagan? Yeah, quite the science powerhouse team. But so she proposed that eukaryotic cells evolved due to a beneficial endosymbiotic relationship between prokaryotes, like bacteria and archaeobacteria. So the idea is that one of the organelles that you find in animal cells as well as plant cells, and pretty much every eukaryotic cell can have one or more, sometimes thousands, like your cardiac cells, they need lots of energy. They can have 5,000 of these mitochondria per cell. And the mitochondria, anyone know what the mitochondria does? Everyone says it's the powerhouse of the cell. Yeah, that's one of its main functions, has several other functions as well. But that's what we're going to be focusing on is how mitochondria have some parallels that warrants the idea that maybe it was an ancient bacteria endosymbiont. And in plant cells, they also have mitochondria, but in addition they have chloroplasts where photosynthesis takes place. And there are some single-celled organisms called cyanobacteria that have some parallels in terms of their overall function to photosynthesis. So let's take a look at a representative type of eukaryotic cell where we've got a nucleus and various organelles, including the endoplasmic reticulum and the Golgi. Here's the relative size of a single mitochondrion, and remember you can have dozens or even thousands of them per cell. So this has some features of bacteria. So maybe one of these got engulfed by the cell and then somehow they started trading genes so that the endosymbiont couldn't leave, because now it was totally dependent on the host cell, and then they were forever captured through all modern descendants, through all eukaryotic cells. And then in a plant cell, which has a thick cell wall, you have these, this is the nucleus, here's a chloroplast, and they have these plastids inside of them that have many important enzymes for photosynthesis. A lot of them are actually embedded in the membrane, but maybe this was a bacteria, a photosynthesizing bacteria that got trapped and formed what are all modern plant cells today. So what is some of the supporting evidence besides the fact that they're about the same size and shape? Well, mitochondria, chloroplasts, and prokaryotic cells share some things in common. Not only are they morphologically similar sizes, but even their ribosomes have similar molecular weight, similar sizes.
Where's the ribosome?
Whoops, let's go back. You can't see them, but there's thousands and thousands of them floating in either the cytoplasm, or there are some actually embedded in the rough endoplasmic reticulum to synthesize proteins that are going to be destined for other vesicles or to be shipped out of the cell. So yeah, they're in this schematic, they're really too small to see, but just imagine there's basically thousands or even millions of these 3D printers all floating around. Some of them are free-floating, we call them free ribozymes, and some of them are embedded in the endoplasmic reticulum.
Just quick. So in this case, then it would be for the bacteria, they would be inside the bacteria, but in this case, they are outside of these units.
Correct. Yeah. In bacteria, since they don't have organelles, they're just in the cytoplasm. Another feature that they all share in common is the double membranes. I'll go back to this picture, and you can see that the mitochondria has an outer membrane that's pretty smooth, and then a highly invaginated inner membrane with these folds, these cristae, that increase the surface area where you find your complexes of electron transport chain that produce 95% of your cell's ATP. And then located within the inner membrane is the matrix, and that's where the citric acid cycle takes place, among other functions. And there's also a very key distinguishing area in between the membranes called the intermembrane space, the space between the membranes, which is where the proton gradient is established that powers those the ATP synthase by the pumping of protons through most of the complexes. I'll show you an animation after this is over that explains that better. So also the chloroplast has an outer membrane, and then these thylakoid membranes, there's basically two sets of membranes. Now with bacteria, they don't have two sets of membranes. They have a plasma membrane, which is just their cell membrane that kind of holds everything in there. It's also where their electron transport chain takes place. Those enzymes or complexes are embedded in that plasma membrane. And then they have a periplasmic space and then a thick cell wall. So the periplasmic space would be like the intermembrane space of the mitochondria, and instead of having another membrane on the outside, they have a thick cell wall. Other common features, well, our DNA is stored in linear strands called chromosomes, but mitochondria have their own genome separate from the nuclear DNA called the mitochondrial DNA genome. And this is circular in most species, not all, but it makes copying it a little bit easier, and I'll explain why coming up. Chloroplasts also have a separate region of where they store their genetic information called the cpDNA, or chloroplast DNA. So that it contains different genes than what are found in the nucleus. And then bacteria, they don't have usually don't have linear DNA. Almost all of their DNA is circular, and sometimes they even have accessory circular DNA called plasmids that might contain information like how to make an antibiotic to kill off their competition, or how to have the they also need to have the resistance for that antibiotic, an enzyme that will break up the antibiotic in their own cell so they don't poison themselves. And they will freely share this information with other bacteria, even other species, through a little pili a little tube that they can share these plasmids with. So yeah, mostly circular DNA, unlike host cell DNA, which is usually linear. Also, the organelles, and bacteria multiply by binary fission. They split in half. This is a very carefully choreographed process because you need to make copies of the mitochondrial genome or the chloroplast DNA before it pinches in half. You don't want to get 70% of it in one half and 30 in the other. So this is all a programmed and timed process, as opposed to fertilization, which a lot of eukaryotic cells undergo, where you have sperm and eggs and cell division that's not necessarily a binary exponential growth. So in addition, plants have chloroplasts that share photosynthetic pathways with cyanobacteria. There are some important differences. So another term for the endosymbiosis theory is the symbiogenesis hypothesis. And the reason they think that this host cell harbors and kept and retain this endosymbiont is that they find crucial genes that code for mitochondrial proteins in the nucleus. And in fact, the mitochondrial genome only codes for a handful of proteins. It needs to get most of its proteins from the nucleus. So they think enough exchange happened where they became interdependent on each other that the endosymbiont couldn't escape without dying. It just couldn't live on its own, kind of like a parasite that mutates so badly that it can't make its own food anymore and has to rely on a host, and it's forever linked to that host because it seems that the degradation of information is mainly a one-way road to loss of information. You're not going to do some kind of revertant mutation that's going to get a parasite back to being free living anymore. We're just going to see more and more parasites as time goes on because of loss of information. So this makes the engulfed cell not able to escape, otherwise it would immediately die because it wouldn't have all the parts it needs to survive on its own. The genes are now somehow inserted into the host nucleus, nuclear DNA. So yeah, they become part of the host genome. Another factor that makes scientists think that maybe this hypothesis is true is when they study the genes of Rickettsia bacteria, they find some similar genes in mitochondria. Also, plastids and the chloroplasts share many genes with cyanobacteria, these photosynthetic single-celled organisms. Other shared similarities, if we look at proteins called porins, which they make little holes in the membranes to allow things to get in and out that otherwise wouldn't cross membranes, such as large molecules. Anything that's polar has to have some kind of transport mechanism to get things across the membrane. Well, we find porins in the plasma membrane of bacteria as well as the outer membrane of mitochondria and of chloroplasts, these channel-like proteins. Cardiolipin, which is a very complex lipid that was first identified in cow cardiac tissue, where there's lots of mitochondria, is also found in the plasma membrane of most bacteria, as well as exclusively in the mitochondrial inner membrane for eukaryotic cells. Chloroplasts don't have cardiolipin, but introns, these are interrupting sequences in, so when DNA is transcribed into pre-messenger RNA, there are some regions in there that don't code for proteins, and they're there for a reason, because if you just willy-nilly delete them, bad things happen. But they allow things like alternative splicing, so you can have a longer segment of a messenger RNA, and through the spliceosome complex, you can make various sort of flavors of the proteins. Let's say you want to, in one instance, you want a protein that's going to be anchored to the cell membrane. So you include the portion that is going to make the hydrophobic amino acids that are going to act as a lipid anchor that hold it in the membrane. But in another instance, you might want to secrete that protein out of the cell, so the spliceosome will exclude that portion of the messenger RNA that codes for the hydrophobic amino acids. So yeah, these interrupting sequences are spliced out by the spliceosome. You don't find these in bacteria, but you do find them in the mitochondria genome of some plants and fungi. And then of course you find it all throughout the nuclear DNA that codes for messenger RNA, as well as chloroplasts. This is also interesting. So normally when translation starts, the ribosome is looking for a three-letter codon called the start codon. AUG is what it's looking for on the messenger RNA. And that also codes for the amino acid methionine. But with bacteria and mitochondria and chloroplasts, that methionine is chemically modified. It's a post-translational modification, or actually, in this case, it would be pre, because the informal methionine would have to be synthesized and attached to a transfer RNA, which is then brought into the ribosome, and then that begins the process of protein synthesis that has this modified methionine, informal methionine. So there's a aldehyde group that's attached to the amine nitrogen, and so that's another thing they share in common. So normally, eukaryotic nuclear genes or even archaeobacteria, they have an unmodified, straight up, plain old methionine as their first amino acid in proteins. Later on, that modified amino acid can be removed, but in any case, this is one of the reasons why they think that they're endosymbionts, ancient endosymbionts, because they share this strange modified amino acid as their initiating amino acid. So now let's get to the criticisms of the theory. One, that's kind of a minor criticism, is that not all mitochondria have circular DNA. Counter-argument might be, well, maybe it was circular at one point and then it became linear, but that poses more problems. I mean, the way that a circular piece of DNA is replicated is quite different than the way a linear strand of DNA is replicated. And it's interesting, even in the same genus of yeast, you can have two species that differ based on whether their mitochondrial DNA is circular versus linear. So linear DNA, the problem with making copies of linear DNA is when this large, complex DNA polymerase scans along the DNA to make a messenger RNA copy, it gets to the end and it can't quite make a copy of the new strand from the template strand. So there's an enzyme called telomerase that puts some sacrificial DNA on the end of the chromosome. And that way, it's okay if that gets shorter because it doesn't code for anything important. It's just like, okay, yeah, go ahead and get shorter and shorter. And eventually in our cells, like not all of our cells keep making telomerase. And so every time your cell divides, the chromosomes get a little bit shorter until you start actually getting into some important areas where genes get destroyed. And that's part of the aging process. It's part of what limits us. Now, rapidly dividing cells like immune cells and the cells that line your respiratory tract or GI tract, they've got a lot of telomerase activity. They're putting on all sorts of sacrificial DNA on the end, so that high turnover isn't going to end up shortening the chromosomes, and you won't have problems. Cancer cells, same thing. They become immortalized because they have high telomerase activity. Their chromosomes aren't getting shorter. So there's a lot of complicated differences in the processing of linear versus circular DNA, especially in its replication. Another difference is that for the replication machinery to recognize mitochondrial DNA, it has to start at a place called the D loop, the displacement loop. In bacteria, their origin of replication is called the ORI site, the ORI. So different machinery, different recognition template. So both mechanisms differ in many different ways. What are the cell membrane differences? We already looked at that there's cardiolipin in the inner membrane, but the outer membrane of a mitochondria is very, very different than the cell wall that you find in bacteria. The cell wall is much more thick, rigid, contains different components, like peptidoglycan that you exclusively find in bacteria, and lipopolysaccharides, things that our immune system would recognize today as being a sign of infection. They can cause inflammation. Other obstacles, well, here's a question. Why would the endosymbiont want to export its hard-earned ATP to the host cell? It takes a lot of work to make ATP. And ATP is large and polar. It's not going to just cross through a membrane. It requires sophisticated transport complex to get the ATP across the membrane once it's made. So how long is that endosymbiont going to sit inside of its host cell making ATP before it can finally figure out how to get it out of the cell for what advantage? Why is it giving up precious energy to the host cell? Those two are just going to end up dead long before they ever somehow work out a partnership. 95% of a cell's ATP, at least 95, sometimes 99% of the cell's ATP is synthesized by mitochondria. So that's a lot of ATP that has to get exported out of this so-called endosymbiont, or out of the mitochondria, to feed that hungry host cell. So what is the mechanism by which the host and the endosymbiont cells exchange genes without causing their destruction through partial copying or gene disruption? Remember, they got to somehow trade enough genes so that the endosymbiont becomes fixed inside that cell and can't escape without dying. And so how is it going to do that? Is the whole gene has to be copied perfectly, not part of the gene, and somehow it has to be integrated into the host nuclear DNA? And is that going to be a random process? Is it going to just stick that DNA right in the middle of a crucial gene and then they're both dead? So there's a lot of unanswered questions here that make this very implausible. Why are most of the proteins needed by mitochondria encoded in the nuclear DNA? Yeah.
As an additional one, it doesn't just need to copy it, but then the host nucleus has to then be able to recognize and read out like it needs the start and the initiation.
Yep, they have the same codons and machinery for transcription, translation.
Proteins that are needed in the mitochondria actually have to have proper targeting sequence in front that
That's right, a zip code.
to mitochondria, not to Golgi or to goodness knows where.
That's right, yeah.
It has to, it's specifically encoded with the target location.
So not only is there a code within a code, that peptide that's synthesized has to have that little zip code on there to say, hey, this needs to go where, yeah, exactly. There has to be receptors that say, oh, I'm the reader of the zip code. So I know to take you
This is the
simultaneously.
Many proteins that are produced have specific pre-pro section, which essentially has a targeting address on it where that protein is supposed to end up.
Right, and a lot of those proteins, once they end up in the right destination, there's an enzyme that will cut off the zip code because they don't want it to interfere with the folding of the protein. Yeah, great point. So what if genes from the endosymbiont were under different regulatory controls than the host cell? That's the issue you brought up. Like, you got to be able to turn on the gene when necessary and turn it off when you no longer need it, or you waste resources, or you do worse, you overproduce a protein that, it's like taking 5 times as much of your prescription medicine. Bad things can happen, right? You got to be able to control all of this. So they need to have the same recognition signals and means of turning on and turning off genes. What if that host cell had some sort of primitive immunity where it only recognized methylated DNA as its own and non-methylated DNA as foreign, and so it starts to reject or destroy the endosymbiont or any of its genes that are trying to become incorporated into its own genome. And then how would mitochondrial proteins synthesized from nuclear DNA even enter through one or both membranes of the mitochondria? Those proteins are large, and they're not going to cross membranes. And the way I have an animation that I'll show you afterwards for those that can stick around that shows you just how complicated the process is of getting the roughly 1,500 proteins needed by the mitochondria, of which almost all of them are made in the cytoplasm by the host, by the nuclear DNA. And some of those proteins need to end up in the outer membrane. Some of them need to be in the inner membrane embedded in there. Some of them need to be in the intermembrane space, and some of them need to go all the way into the matrix. So there's multiple signals for multiple destinations, multiple types of transporters. Yeah.
It turns out that bacteria use different dialect to encode for the same amino acids than eukaryotic cells use.
Yeah, codon table is not 100% universal.
Yes. When you have different dialects in different organisms, yeasts use different dialects from bacteria or eukaryotic cells. So when you have a sequence that comes in which is in a different dialect, that sequence is recognized by restriction enzymes that chop up that sequence.
That's right.
Because it's not our language. Even though it theoretically would encode for perhaps the right protein, it's just not going to be recognized as welcome.
Yeah, that's a good point. For those of you that don't know what restriction endonucleases are, these restriction enzymes are sort of a straightforward way of bacteria to protect themselves from foreign DNA, like a viral DNA insertion event. So they're enzymes that look for particular sequences. They scan the DNA, and these sequences are usually a short palindrome, and they chop that DNA and say, hey, this is foreign, we don't want it here, let's cut it up. So endonuclease means it chops interior to the DNA as opposed to cutting up the ends when you want to recycle the DNA that you're eating. So this is a problem if the chemical tags to the DNA don't tell the restriction enzymes that, hey, don't digest this is self. And 2, if you all of a sudden introduce a sequence that has a restriction site that the enzyme now recognizes and cuts up as being a foreign invader.
Yeah, and importantly, not all of the systems that would interfere with this are even necessarily like immune. Like the cell itself has its own spell checking to
Right.
make sure that you don't run up and make a useless protein because you ran nonsense through it. And that would, if they are using a different coding system, then that would get caught and chopped up, not even necessarily as an immune response, just because it's wrong.
Yep. Yeah, or if it doesn't fold properly, has the same fold assisting proteins. Say, hey, we don't recognize this. Yeah.
So you were saying of the 1,000 to 1,500 proteins made by mitochondria, only 9 or 13 are encoded by mitochondria.
Yeah, and this is interesting. Why is it different in yeast versus humans if they came from the same endosymbiont? Or was this a co-option event that it happened one time in one cell and those went on to become yeast, and then which are eukaryotic cells, and then happened another time where 13 of the genes were retained in the mitochondria, and then why in the world would it send out, in the case of humans, about 1,500 genes for proteins to the nucleus just to make sure that it can't escape now because it's so utterly reliant. Like 99% of its proteins are now made by the host cell instead of its own mitochondrial genome.
So are these 9 and 13 that they will say is the same code that you find in bacteria and
Most of these are actually proteins that are involved in the respiratory chain complexes, so the electron transport chain complexes. But there are some differences in the proteins. But yeah, in the case of these 9 or 13, they do share some similarities in terms of what they're coding for.
is this the same proportion in other organisms as well?
This is just the one paper I found that happened to mention yeast and human, so I assume there's some variability. But yeast, they're almost closer to bacteria, except they have organelles. They're single-celled. I mean, they can grow in chains as well. But yeah, so there is some variability in there, which I think is interesting. But what's really interesting to me is how in the world did it export that many crucial genes to the host nucleus? And then, of course, if 1,500 proteins are being made in the cytoplasm by the host cell, you've got to get all 1,500 varieties of proteins back into the matrix, or into the outer membrane, or the inner membrane, or the inner membrane space. So these carriers that allow these proteins to go through, and transporter mechanisms, and recognition sequences, and destination processing apparatus, the apparati are very complicated. So I'm going to end, because some of you have to leave. We're going to end the lecture now, and then we'll have Q&A. And then for those that want to stick around, I got two short, 3 to 5 minutes animations that show the complex process of trying to get proteins that are made in the cytoplasm imported into the various parts of the mitochondria. And one of them will be a schematic diagram that gives an overall picture of how it happens, and I'll show one that's more realistic that shows the controlled, what looks like chaos, and even that is a very simplified CGI animation of what's going on in the mitochondria. So we'll take some questions right now.
It seems like this whole hypothesis is, like, unless you were trying to make evolution work, you would never think, oh, let's hypothesize that this cell was formed in this way.
Right, and part of it is just, we have anaerobic bacteria that can go without oxygen, and they'll eke by on enough ATP made by glycolysis and basic fermentation of small molecules. But the demands of ATP in a eukaryotic cell are tremendous. I'm 6 feet, weigh about 200 pounds. My body every day is making about 200 pounds of ATP a day. And then it dephosphorylates when all those various, every time I breathe, move a muscle, there's all sorts of reactions that need the phosphate off the ATP to phosphorylate another protein. I mean, there's just so many things that use ATP in your body that you're constantly having to create ATP, and then it loses a phosphate and makes ADP, and then rephosphorylates. And so, you have maybe 1 gram excess of ATP compared to ADP, the less energetic form of it when it loses a phosphate. And the mitochondria are doing, like I said, 95, 99% of your ATP synthesis.